In Chapter 16, we're going to be talking about regulation of gene expression. And we'll talk about what does that term mean? What does it mean to regulate gene expression? And I am going to deviate a little bit from the order the textbook goes in. I'm going to start by talking about regulation of gene expression in viruses, because that was where we really first learned about this whole idea. And from there, then we'll talk about, well, what does this phenomenon look like in prokaryotic cells, and then go into eukaryotic cells and a couple of more advanced ideas that we've studied in eukaryotes from there. All right, so to start this off with, what does that term mean, regulation of gene expression? And so gene expression, in a nutshell, is just referring to the fact that a gene contains the instructions to produce a certain polypeptide, right, a certain protein. And so we would say a gene is expressed if the protein that's coded for by that gene is produced in the cell. And so that means we're going through the whole central dogma here, right? We transcribe the gene, process the RNA, and then translate that messenger RNA out in the cytoplasm to make that protein. So that protein's there doing its job, the gene is expressed. So obviously here we're talking about how that process gets regulated. And as I mentioned on the first slide, researchers first came across this whole idea of gene expression through studying viruses. So the textbook doesn't really know exactly what to do with viruses, and that's kind of understandable because they do fit in with biology in that they affect living cells in a big way, but they're not technically living organisms. They're really cellular parasites, essentially. So we get a little bit of virus biology here and a little bit there. So we'll kind of deviate and just take a quick look at the virus life cycle. But essentially, you can think of a virus as basically a parasitic piece of DNA or RNA. So it consists of a small genome, either a DNA or RNA genome, a few proteins, and the virus's whole role is just to get inside of a host cell and then hijack that cell and use the cell's metabolic machinery to copy itself, and then move on to the next cell. So let's just take a quick sidestep here and look at virus life cycles. And we've got two common types of virus life cycles, what's called the lytic or the lysogenic cycle. And just to give you a quick overview of what the difference is between those, a lytic cycle would be one where a virus is infecting a cell and then immediately producing new virus particles upon infection. So the example you have here is a bacteriophage, which is a virus specialized for infecting bacterial cells. The way bacteriophage work is they attach to the surface of the bacterium. Bacteria is in green here, phage is in orange. Injects its genetic material, DNA or RNA, into the host cell. For the majority of bacteriophage, they have DNA as their genetic material. And then once that DNA is inside the host cell, it gets transcribed and translated into virus proteins that then assemble into new virus particles. You can see those forming inside the bacterium here. Eventually, they would just fill up that whole bacterium and come bursting out and then move on to the next cell. So that would be the lytic cycle. And in the interest of time, I'm not going to go through the video, but for example, the flu virus operates on a lytic cycle. The coronavirus also, for that matter, as soon as they infect your cells, they begin producing new virus particles and then move on to the next cell. So that is in contrast to what's called the lysogenic viral life cycle, which can be seen here on the right. And in the lysogenic cycle, either the virus or the virus genome gets inside the host cell. In this case, we're back to bacteriophage, so it injects its genome into the host cell, DNA in this case, and instead of immediately producing new virus particles, that viral DNA just hangs out. And in this particular case, it actually integrates into the bacterial chromosome, so essentially becomes part of the bacterial genome at that point. And now every time this bacteria divides, it's going to copy that viral DNA along with its own chromosome. So you would end up with, hundreds or thousands of bacterium now that have that viral DNA inside of them. And then at some point, as you can see in this cartoon here, um, we may switch on transcription and translation of those viral genes, and then at that point we would enter the lytic cycle. So we could go from the lysogenic into the lytic. And what that trigger is in some cases may not be known, but this is kind of scary because it just means the virus can kind of hide out, lie in wait, and then at some point down the road, all of a sudden basically kill all of those cells essentially simultaneously. There is a special term for what's going on here in this lysogenic cycle when the viral genome is integrated into the host chromosome, like you see here. At this point, this would be considered a provirus, so that integrated genome. Something similar would happen, for instance, with HIV in human cells. HIV integrates into human chromosome, and at that point is a provirus, and you would be HIV positive. Similar idea. Okay, so let's go back to this idea of gene expression. So, what did we understand from studying viruses about this whole idea of gene expression? And as I said before, we really learned some of the just basics. So, for example, we learned that in order for genes to be expressed, they need a sort of companion sequence called the promoter, which basically sits in front of every gene. And the promoter, which is shown in essentially in green here, would be a site where RNA polymerase could bind and then begin to transcribe that particular gene. So we got this basic idea, okay, every gene needs this promoter. The promoter is the DNA sequence where RNA polymerase binds and starts transcription. But then we also discovered, okay, there are ways we could either enhance or repress transcription through what we would call either positive or negative regulation. And that would typically involve the activity of either enhancer or repressor type proteins. So for example, in image A up here, you see this repressor protein in green comes in and binds to the DNA and blocks transcription from happening. So RNA polymerase would no longer be able to bind to the promoter and successfully transcribe that gene. But you could have just the opposite type of situation where you could have some sort of enhancer type protein that comes in and binds and actually helps RNA polymerase work more efficiently. So you would actually get more transcription or faster transcription in that case. So we could kind of fine tune the amount of transcription through either negative or positive regulation. So those are kind of the basics that we learned from viruses. And of course, once we had that basic understanding, researchers wanted to know, oh, well, how does this work in actual living cells, in prokaryotes and eukaryotes? So what you're going to see is the same basic principles still apply, but things get arguably more interesting and a little bit more complicated. So we'll start with prokaryotes and see what regulation of gene expression looks like there. And prokaryotes are unique because they use what's called the operon system. And if you have any OCD tendencies, this will appeal to you because the operon system is very organized and sort of a neat and tidy way to organize and regulate gene expression. And so we're going to look at just one example of an operon that's called the lac operon, and it is so named because it is involved with regulating the genes that are important for digesting lactose, which is a sugar you find in dairy products. And even though we're looking at this one specific operon, just keep in mind that there are many others, and they are all sort of organized and potentially operating in a similar way. So what you see here is you'll have the genes that are part of this operon, and it's always going to be multiple genes. In this particular lac operon, you have 3 genes that we're calling the structural genes, but these basically code for 3 different enzymes that are all important in that process of digesting lactose. So this comes back to the organization I was talking about. We've got 3 genes that are all involved in the same biological process grouped together here in the genome, just sitting right next to each other, back to back to back. And then there's going to be just a single promoter for all 3 of those genes. And remember, the promoter is where RNA polymerase would bind if it wants to transcribe these structural genes. And then you'll also notice there's this DNA sequence between the promoter and the genes called the operator. And this is sort of where that term operon kind of ties in with this idea of operator. And this is really a regulatory sequence. And I'll show you what happens at the operator here in just a second. So all of that would be considered the operon in this case. The promoter, one promoter, an operator, and then our structural genes, 3 in this case. You also have these related genes right next door that are technically not part of the operon, but they do play into the whole story here, which is this gene encoded by this yellow stretch of DNA here that makes a repressor protein. And that gene actually has its own promoter and regulatory region there. So this gene would be transcribed from this promoter. These 3 structural genes would be transcribed from this promoter here, part of the operon. All right, and this was just giving you a little bit more detail on the names of the enzymes that are encoded by these 3 structural genes, but I'm not so worried about you knowing those names, as long as you're aware that all 3 of the proteins coded for by these genes are important for digesting, breaking down lactose, so the cell could use that as an energy source. Okay, so how does this whole system work? And it's really cool because it's sort of an automatic on-off in terms of turning transcription of those genes on or off. And you can imagine, so let's say you are a bacteria, an E. coli, and you're going about your business, and there's no lactose in your environment, right? No lactose for you to digest, then it wouldn't make sense for you to produce those 3 enzymes, right? You're basically just wasting your time and energy making enzymes that you don't need. So in this situation, if there's no lactose around, the bacterium won't produce those enzymes. Transcription should be off. But if lactose shows up, then we would want to turn transcription on so we could make those 3 proteins, digest the lactose, and make use of it. So the kind of the default state here is no transcription. We would say transcription would be quote unquote off in this case. And we have to actively turn it on, induce it. So this would be considered an inducible operon. And there are other operons whose default state is to be on. They're like always being transcribed, and you would have to come in and actively turn them off. Those would be considered repressible. But we'll just focus on this inducible example here. Okay, so how does this work? Well, what's happening is, if you remember the gene that was right next door to the lac operon that I mentioned, the yellow gene here with its own promoter, this one is constantly being transcribed and translated, and it produces this repressor protein. And what the repressor protein likes to do is to actually bind to that operator DNA sequence that was part of the lac operon, right between the promoter and the structural genes. And so because this protein is always being made, it's always lurking around, as soon as it sees DNA combined to, it will bind, and it just sits there on that operator sequence. And that is going to have the effect of keeping transcription turned off, because if RNA polymerase, which is the pink blob here, shows up and says, hey, there's a promoter and some genes I'd like to transcribe, it's going to get turned away. It just physically bumps into that repressor protein and it can't do its job. So no transcription means we do not make the enzymes coded for by these genes. And that is the default state when there's no lactose around. Transcription turned off. So how do we turn it on? Well, we need to induce this system, and we induce it with lactose. So if that lactose sugar shows up, what it will do is it will physically bind to that repressor protein and cause it to change its shape. So now the repressor, you can see it sort of crossed its legs. Let me go back a slide. Right. Crossed its legs, so now it can't bind to the DNA anymore like it was. So it falls off the DNA. And now RNA polymerase can bind and transcribe, right? So as soon as lactose showed up, we took away that inhibition, and RNA polymerase transcribes all three of these genes as basically one messenger RNA. It's technically called a polycistronic messenger RNA. And then that one messenger RNA gets translated into the three different enzymes. So we turn the system on, right? And so, if we follow this through, well, then now what's going to happen? These enzymes are going to digest lactose, right? So they're hunting around for these lactose molecules. As they find them, they break them down so they can be used for energy. And as the lactose goes away, what you're going to see is, well, now there's no more lactose around to bind to the repressor. So the repressor proteins go back to their sort of natural state here, bind to the DNA again, and the whole thing turns itself off. So that's what I was trying to get at with the kind of automatic on-off. If lactose is absent, no transcription. If lactose shows up, transcription is activated until all the lactose is used. So it's really quite an elegant system, I think you could say. And very, very tidy, very organized, right? You have all these genes involved with the same processes grouped together in these different operons inside the bacterial genome. And just to add one more slight layer of complexity to this, we've been talking about the repressor protein and how it keeps the lac operon turned off. Well, there's also another protein that can interact with this operon called the CRP protein, being shown here, and it is actually an activator. So the lac operon was under some negative control by that repressor, but we're seeing here it's also under positive control by this CRP. And so basically the CRP only gets activated when glucose levels are really low, lactose is high, then this protein will bind to the promoter and kind of supercharge the RNA polymerase. So it can basically make the most of the lactose that is present. Yeah, so I think I already said that. All right. So we've got these two layers of regulation, both positive and negative. And as I mentioned before, just keep in mind, this was an operon that was off by default and we had to induce it, but there are others out there that are on by default and we have to repress them, so repressible operons. And if you're a biology major, if you take genetics and or cell biology down the road, you'll see some other examples of operons that might be considered repressible. I'm going to stop there and we'll